thank you first and foremost for your invitation. It's an honor to be on your speaker schedule. I have a sweet spot in my heart for all things British, and many years of relationships and experiences in your beautiful country, culminating in a 2-year sabbatical with Giles Budge, who is, I understand, in the audience tonight. Hello, Giles. And I was with Giles for the 2012 and 2013 seasons in a project that rather kicked off the subject that I'll be talking about tonight, which in my opinion is a grossly overlooked aspect of bee biology and management. If your beekeeping books are like ours, there is a large canon of respected texts that talk about the biology of honeybees and the breeding of honeybees. And the ones about breeding, they have almost an apologetic sound to them when they talk about the queen's habit of multiple mating. And you can appreciate this because these books will explain how, as a bee breeder, you should exercise some kind of selection against a population of colonies for the character that you're interested in. And then you should propagate from those queens whose colonies express the character you want. And you may enjoy some measure of success with this method, though it is shockingly crude and imprecise. And these books will continue to say that you will graft larvae from these strong producers and then expose these virgin queens to essentially an unmanaged element. And you basically hope for the best. And it's, oh yes, the queen will mate with multiple males, and in the process, it probably will dilute some of your breeding efforts, but gosh, that's just the biology of the animal, and it's just the way it is. So there's this apologetic attitude that these old books bring to bee breeding. And I am wanting to contrast that, classical breeding approach with what I think is more pertinent for the biology of the animal and also the management of the animal. And ask ourselves, how do honeybees use genetics to solve their problems in their natural history? And I think we'll find tonight that it's very different from the approach that I just described to you that is sort of mainstream bee breeding. So this is interesting from the aspect of the honeybee's evolution, but it is extremely timely in the day and age we're living now, where we have many different types of stressors that are hazarding our bees and their health and their productivity. And it's no secret that the holy grail that we all pursue is a bee that is genetically resistant to many of these stressors. Nobody likes using chemicals inside beehives. So a bee that can handle it, with its own genetic resources is obviously, to be preferred. Okay, so this is going to be rather a big picture talk tonight, but I promise you, I'm going to bring it down into some principles that I think are very grab-onable. You can take it home with you tonight and improve your beekeeping with. So just bear that in mind. We're going to end up in a practical place. Well, what you're seeing here is a phylogeny of honeybees on planet Earth. I mean, darn it, you don't get much bigger than this. And I'm not going to go through it in detail. Please don't panic. But there's a couple high points here that I want to mention to you. First and foremost is right out of the gate. Those top 3 branches you see on the top left document what is thought to be the 3 main domains of life: the archaea, the primitive single-celled organisms, then the eubacteria, the eukaryotic single-celled organisms that have nuclei in their cells, and then the eukarya, which is all the rest. That's you and me and the plants and the fungi. These are the animals, the multicellular organisms. Well, I was reading just last week in my, right here, the latest issue of National Geographic magazine. And it was explaining how there's some conversation that there maybe ought to be a 4th domain of life. That's right, the viruses. because they are so hard to pigeonhole into any of these three categories. They don't really easily fit in any one of them. So we may have a fourth domain someday. But the point for now is that we and the honeybees belong in the eukarya. We are the multicellular organisms. Now, the interesting thing about us and honeybees is that honeybees are technically one order of biology higher than us. If you think about it, our bodies are made up of eukaryotic cells. They're individual cells, each of which has its DNA encased in a nucleus, and essentially are more or less independent because any given eukaryotic cell can perform most of life's functions. It can acquire nutrients, it can reproduce, it can excrete waste products. So a eukaryotic cell is pretty darn close to an autonomous entity. And then the organisms that it makes up, like you and I, are autonomous entities. Well, what do the honeybees do? They have autonomous entities too. We call them worker bees, and they come together to form a higher unit, which we call the superorganism. So on the scale of evolutionary complexity, humans, ironically, occupy a penultimate position, and honeybees are above us. I don't know about you, but I find that poetic sometimes. There may be a lesson there for us, isn't there? Well, I'm going to cut through several hundreds of millions of years now, and we're going to jump down to the bottom row. On the far left, you see the aculeata. These are the constricted waste hymenopterans. Okay, this is the ants and the wasps and the bees that have that constricted waste, and the aculeates are technically hunters. These are wasps that are carnivorous and predatory, and from them evolved the bees. And take a look down at the bottom left at about the 7 o'clock position. You'll see that green zone spanning roughly from 140 to 100 million years ago. And this is the arrival of the flowering plants. The flowering plants, and then immediately after the flowering plants, we see the emergence of, you guessed it, the true bees, the Anthophila. So bees are essentially vegetarian wasps. These are hunting carnivores in their ancestry who took advantage of this new source of protein that was showing up in the environment in the form of plant pollen. And these vegetarian wasps became really good at it. And they evolved adaptations like branched hairs for collecting pollen and corbicula on their legs for transporting pollen. And it was also a very rich group for the evolution of sociality. And moving far ahead now to about the 5 o'clock position, you see that pink zone there? That is the evolution of true sociality. And we're getting pretty close to the honeybees at this juncture. In fact, so much of the evolution of honeybees that's of interest to us as beekeepers is crammed into those final little divisions that you see on the bottom right. And in fact, I had to truncate the time scale to make it work. It's so recent. Because by the time we have the divergence of honeybees into their different lineages and their races, we're talking in a scale of tens of thousands of years. So this is really, really recent stuff. So this is a long history. The lineage of honeybees, like for all things on Earth, goes back to the very beginning of life. And ultimately, we really are all connected, not just proverbially, but literally. Okay, advancing slides here. There we go. Okay, here we have a map of planet Earth, and I want to just throw out there to you all a theory that's out there. Okay, this is a paper published about, it's not so new anymore, it's about 2013, I think it came out, and it is an hypothesis that I think every beekeeper should know about because it raises a really interesting possibility. We all have known or thought or believe that the southeastern Asia is the origin point of the genus Apis. And there's a good reason for that. Generally in evolution and in biogeography, we think that an area that we find the most variance of a particular group, where we see that most the variance occurring, is probably where that group originally evolved. And as variants move away from that epicenter, there's sort of a dilution effect as you go out from that point of origin, so that line becomes less variable as we go away from that center of gravity where the group evolved. And so Asia, Southeast Asia, is kind of a no-brainer by that criterion, because that's where we see the greatest numbers of species of Apis to this day. However, in 2013, there was a very compelling alternate explanation that was published, and it was based on fossil evidence. And it just so happens that the zone of the oldest fossils of the genus Apis don't occur in Southeast Asia. They occur actually in Central Europe. That's right. So these authors are positing that the whole genus actually is natural to Central Europe. Now you notice the globe looks differently there. This is 30 million years ago. And continuing with this scenario, these authors are saying that there's at least one or possibly two lineages of ancient Apis that then immigrated into Southeast Asia. And from there, they radiated into all the variants that we know today. Well, for those of us from South America, there's a little side story to this same paper that we find very satisfying. And that is that as Apis moved into Central Asia and modern-day Siberia, they actually crossed over Beringia into North America, where it left behind in shale from the state of Nevada in our extreme west a fossil of a true Apis. So yes, the genus Apis did make its way to the New World, and these authors named it Apis nearctica. Aniarctica, of course, went extinct, but it had a good run for its money for about 8 million years, where it is thought that climate changes became unfavorable to it. So that's just an interesting coda just to throw out there. I usually make a big deal of that to my North American audiences and let them know that they can take pride in knowing that Apis is in fact native to the New World. But of course, that one went extinct and left the continent empty until historic times when European colonizers brought the bee mellifera into the New World. Okay, back to Central Europe. There was later another branch that moved into Iberia and crossed the Iberian Isthmus into modern-day Africa, and it's this CM lineage that is posited to be the forebearer of what became Apis mellifera. So mellifera is in fact a child of Africa. This now is modern distribution of two species of great significance, mellifera and its sister species, Apis cerana. I think most of you are familiar with this. This is their current natural distribution. Of course, they've since been moved out of this, but this is the natural range of these two important species of Apis. And I just want to pause right here and say this natural distribution should puzzle anybody who is a student of honeybees for a couple reasons. Why are they so distinct from one another? There's no gradation, okay? There's no gradation from Southeast Asia without interruption across west into Africa. No, they're discontinuous distributions. You got cerana here and you got mellifera here, and their natural ranges did not even overlap. And I just, as a student at LSU, I always thought that's weird. That's strange. Why would this one outlier of a genus be out here totally on its own? Well, that's one question that's out there. And then a second question in modern times is equally pertinent, and that of all of the Apis, why does mellifera alone not have its own Asiatic mite? That too, I found a great oddity. That's not the case in Southeast Asia, where there is a comparably variant supply of mites. And a lot of these mites are very specific to their particular apis host. And it's not at all unusual. And these mites and these bees have co-evolved relationships where they're not really parasitic, they're almost more commensal. Very little harm is practiced to these hosts. But mellifera doesn't, has no mite, no Asiatic mite. I always thought that was strange. Well, this model explains both of these two puzzles. If this model is true, then it offers a really good explanation for these two modern phenomena that we witness. And I think more to the point for beekeepers, I think it gives us a strong context to just understanding what we are asking of Apis mellifera when we enter into mite resistance breeding schemes. Because we have to realize that mellifera is not just short on genetic tools for resisting Asiatic mites, it's non-existent. There is no history of mellifera at all. However ancient we take the genus, it has no history in Asia. And so that just gives us context to understand that we are asking mighty things of Apis mellifera when we try to bring it under the influence of our selection programs for Varroa mite. It is an utterly alien parasite to our bees. So this should give us context. It should give us comfort, in finding how hard it's been to breed a resistant mellifera. Now, that's not to say that they don't get resistant. And a good case in point is hygienic behavior. Hygienic behavior is a natural behavior to mellifera, but it evolved in response to bacterial and fungal brood diseases. So when we approach Varroa sensitive hygiene as a tool for mite resistance, we are expecting mellifera to use these tools to new purposes. Okay, so this is a tough demand, and with breeding and with selection, both natural and artificial, mellifera seems to be doing that. Okay, we do get some action of hygienic behavior, but it is putting a tool to a new use. So it's a very hard and weighty request that we're making of mellifera to do this. Okay, so specialist breeding for characters is fighting an uphill battle in this case. Okay, things are interesting for us as beekeepers back on the continent of Africa. And again, now we're looking at a scale of about 300,000 years ago when African ancestral mellifera began a series of radiations out of Africa. The oldest branch, since called the M lineage, reversed its earlier course and went north again across the Isthmus of Iberia back into the continent. And this we call the modern day M lineage. And then the C lineage, about half of the distance ago in time, about 165,000 years ago, emigrated east over the Isthmus of Suez, up into Anatolia and around the Black Sea, entering into Europe from the east. And the C lineage gave rise to the modern day Carnica and Ligustica. Enter glaciers. Okay, glaciers are a big deal for students of honeybees, because we can have the glaciers to thank for some subsequent racial distinctions that evolved. At the last glacial maximum, we had glaciers also covering the Pyrenees and the Alps. And those over the Pyrenees reproductively isolated the M lineage so that it diverged into Apis mellifera iberiensis, the Spanish bee, and Apis mellifera mellifera, the bee of Northern Europe, including the British Isles. You also may want to be reaffirmed that Apis mellifera mellifera is in fact a natural member of the British Isles. This was controversial a few years ago, and Norman Carrick, one of your countrymen, published a very satisfying article in Journal of Apicultural Research, where they did not resort to biology or phylogeny. They resorted instead to good old-fashioned human anthropology and excavated some refuse pits of old pre-Roman encampments in Britain and found residues of beeswax and honeybees. So the Romans did not import mellifera into Britain. Mellifera was already there on its own. Meanwhile, to the south, the Riss glaciation covered over the Alps, and this caused a similar reproductive barrier, causing the C lineage to fractionate into modern-day Carnica and the Italian Ligustica. Well, across that grand panoply of time that I have just spelled out for you, mellifera evolved from a solitary bee into a simply social bee, and by that we mean a female mating to just one male. And these simple eusocial colonies tend to be behaviorally very simple. They don't have a big repertoire of behaviors, and they also tend to have much smaller populations. We have members of them in the family Halictidae today, the sweat bees, that fit this bill. They're very small, have a population maybe measured in the tens. But then the lineage gave rise to the honeybee today. The mothers began practicing multiple mating. And this had the effect of lowering the average relatedness in the nest, which at first thought it would be a bad thing, right? Because the whole idea of social evolution is that you have this close interrelationship, this familial cooperation by staying together with close kin. And if the mother is mating with multiple males, you don't have close kin anymore. You've got a nest made up of a bunch of half-sisters, and it drops that average relatedness precipitously. So what's the advantage? Well, the advantages are huge. The advantages are huge at the level of the colony. At the level of the colony, this practice of multiple mating gave many benefits. The colonies evolved greater complexity, which gave rise ultimately to greater size and greater penetration of their habitats. Apis mellifera is all about genetic diversity. In fact, I'm going to tell you how about genetic diversity Apis mellifera is. I've already told you, one, the queen practices multiple mating, and by doing that, she guarantees a colony that's going to have at least some degree of genetic diversity, and she stores all of that sperm in her spermatheca. You'll be interested to know that I was giving a presentation about polyandry, I think it's been 2 years ago now, to the Devon group. And Glenn Davies is a member there. And Glenn said, oh yeah, we know all about that. Says, we have a group that dissects the spermatheca out of queens and we count the sperm. And I'm shaking my head, really? He said, oh yeah, one of our members is a veterinarian. And so we just use his lab. And Glenn proceeded to send me this image. Okay, so this is an image from Glenn Davies, and it's showing a spermatheca. It's been excised into two here. And the cell wall of the spermatheca is only one cell layer thick. Each of those little black dots you see around the outside is the nucleus of a spermathecal cell, comprising the whole wall. And that mat of yarn, looks like a ball of yarn inside, is all the tails of the honeybee sperm. The honeybee male has sperm of the longest known tail in the animal kingdom. And yes, they do all cram together in that spermatheca, and how they remove themselves without being tangled, I consider one of the great mysteries of life. But they do. Each queen, each egg that passes down the median oviduct, it passes by the spermathecal duct, and on average 6 to 10 sperm are released for every egg. And they remove themselves from entanglement and get the job done. So that's one way that the honeybee queen practices diversity. Another way she does it is drone congregation areas. And I have images of your countryman, Carl Scholler, over by Wales, the border between Wales and England, Hay on Wye. And Carl made a life habit or life hobby out of tracking drone congregation areas with a big cane fishing pole and a queen lure dangling from the end of it. And he would walk his neighborhood, which was very near a Roman encampment, and the marks of the Roman road are still visible there. And he would track these drone congregation areas for decades. It was a life hobby of Carl's. And we know from work on the continent that these drone congregation areas can contain drones from a shocking number of colonies, over 200. Over 200 colonies can be represented in one drone congregation area. Another way that the honeybee practices radical genetic variance is in the formation of their gametes. This is a process called crossing over, which occurs in the testes and the ova of eukaryotic cellular or sexually reproducing animals. The homologous chromosomes will line up with each other, you see on step A, and then at step B, they will cross legs and they will break off and exchange segments of the chromosomes all the way down to D. And then those pairs pull away from each other and then divide yet again and form the individual sperm or the eggs. So this is a radical reshuffling of the dice in the formation of their gametes. And the honeybee, another distinction in the animal kingdom, it has the highest rates of recombination known. So the honeybee uses, well, I lost track. Is that three or four different levels, each of which is an outstanding bid to achieve the end result of a colony that's genetically diverse. Oh, there's actually another one. This is worth telling you. The drone, you know, if you try to ejaculate a drone to collect their semen, you know all about this. There's two horns of the endophallus that come out, and those horns are bright orange. Well, it appears that in a drone congregation area, drones are attracted to these mating signs left by earlier males, which is odd because in most animal kingdom, there's strong male on male competition. Just like the male carpenter bees that I have flying out my window right now. They like to chase each other off. But in the honeybee male, the reverse is true. They cooperate with one another because these orange horns that are sticking out from the mating sign left after a queen mates attracts other males. And it makes sense when you think about it. If the colony is more robust, if it is genetically diverse, then it's in the selfish interest of the drone himself to cooperate with other drones to produce a queen whose colony is very genetically diverse and apt to be very fit and survive. So diversity, suffice it to say, is important in the honeybee. There we go, there's Carl going about his thing with his cane pole. Okay, so what? We got diversity, we got it. Well, what does that mean for the colony? Well, you've probably seen this relationship before. This is an upgrade from Mark Winston's famous table that he published in 1987. I'm in the process of revising this for a book I'm working on. And this, what you're seeing here is a meta-analysis of the average age of workers as they perform these different tasks. You see along the bottom there, we have the increasing age of a worker. And along the left, we have a series of tasks. And suffice it to say that the typical movement is from interior brood care tasks to more exterior tasks like guarding and foraging. Foraging being the terminal task. Nobody survives foraging. That's the ultimate, the final class. Now I notice too that there's a ginormous amount of overlap. Huge amounts of overlap. So these are far from precise values. And this underscores how variance works in the colony. A colony, for example, that loses a large fraction of its forager force, some of the younger ones are able to precociously jump ahead and perform foraging tasks. And the reverse is also true. If a colony loses a huge fraction of its brood population, some of the old ones can regress back to earlier tasks and actually regenerate their mandibular glands and return to feeding larvae. This is true pretty much of all workers, but it's also true that inside these individual tasks, there are specialists. There are individuals who linger longer than average at one particular task. And in the literature, there's even examples of individuals who carry this to an extreme, and they spend their entire life doing just one task. Now, admittedly, this is rare, but these extreme specialists also tend to parse down to the level of an individual subfamily. In other words, they are subfamily based, which is another way of saying they are drone based. They are based on the patriligne from a particular drone. So a queen who accumulates more drones is going to have more of these favorable combinations of specialists, which we hope and think can add up to overall superior performance of a task and ultimately colony fitness. I do want to say in an intellectual honesty that there's also examples where this can go awry. There is examples where over-specialization can actually be counterproductive to a colony's fitness. You may have specialists getting in each other's way. Too many cooks spoil the soup, that kind of a syndrome. So there is a possibility of confounding one another if you have too many specialists. So we're not talking about an infinitude here. We are talking about combinations of specialists that are optimum, not necessarily infinite. Well, with instrumental insemination, we can test and experiment grand ideas like this. I have laid out for you two rather different ways honeybees can use genetics to solve their problems, or to put that more precisely, the way genetics can be used to solve honeybee problems. And one of those is nature's way, right? You just have generation after generation of selection for the peculiarities of that habitat. And yes, there will be strange years where many of the members of that population fall off the cliff. It was maybe a really, really, really brutal winter and the colony is really hammered. All colonies in an area are hammered. Or there may be really liberal seasons where everybody thrives. But over time, we have an averaging out of these extremes so that a population overall is pretty darn fit for the extremes that habitat throws at it. That is not true of artificial breeding schemes that we humans do. We humans generally approach breeding with a particular thing in mind. Honey production. It's all about honey production. We're going to breed for honey. We're going to breed for gentleness. We're going to breed for Varroa mite resistance. Okay? And so generation after generation, for the limited lifetime of a human bee breeder, we succeed more or less. But in so doing, we are blind. We're literally blinded to other characters of equal or greater importance in the colony that we are losing, not actively, but by neglect. And this is another way of describing what's called a genetic bottleneck. When a population is squeezed through a really tight stricture, like a human breeder who's selecting for Varroa-sensitive hygiene, that population will lose other genes. It's just attrition by neglect. And so that's exactly what we do in queen breeding, isn't it? What I just said is exactly what we do. We're putting them through a bottleneck with our particular narrow interests in mind, and we're proceeding blind, not knowing what other important tasks are being lost in the process. So is this an intractable impasse? Is there any way to reconcile these two dynamics? I would like to think there is, and I'm going to present to you now results of a paper we are trying to get published as we speak. I was working today on the reviewers' comments, so this is a paper that's soon to come out that builds very naturally upon the work that Giles and I did back in 2012 and 13, using instrumental insemination to create different levels of polyandry. So the question here is, how does this interface with the human scheme? And let's just use Varroa-sensitive hygiene as our model. And this is a character that is general to many things. It applies to American foul brood, chalk brood. It applies to wax moth eggs. It applies to small hive beetle eggs, and it applies to Varroa mites. And the way this can be tested in the field in a proxy sort of way, so you don't have to infect your colonies with these pests, is to use liquid nitrogen. We use polyvinyl chloride pipe, plumbing pipe. This is an old picture showing steel flashing. But the point is, you screw this cylinder into an area of sealed brood, count the number of skipped cells, and then pour in liquid nitrogen, which in a matter of minutes freezes that circle of brood as solid as a rock. And then you put that freeze-killed brood back into the colony and come back after 24 hours and figure out the percentage of cells that had been removed. The colony on the bottom right is a rather mediocre level of expression, whereas the colony on the bottom left has removed 100% of the freeze-killed brood. So if I were a breeder, I would propagate the queen from the bottom left. And this is what we did, using 2 lines of drone semen, wild-type drones who were completely unselected for any kind of mite resistance character. And then we used 2 levels of polyandry, low and high. I replicated this experiment with my colleague, Dr. Debbie Delaney at the University of Delaware. And yes, my students had a lot of fun with Debbie Delaney from Delaware with Keith Delaplane. That's a lot of D's. And well, let's just go ahead and show you here what we did. We had these 2 levels of interest, right? Polyandry, low and high, and previous selection, VSH or wild type. And so you can see by following that matrix that we created every possible combination of those 2 main factors that we were interested in. And this is called a factorial design. It's very common in agriculture when you're trying to rapidly get information about how 2 or more levels interact with one another. In Delaware, Debbie had rather poor survival of her drone source colonies, so we unfortunately had to limit the spread of our polyandry to queens inseminated with either 1 drone in Delaware or 4. In Georgia, where the weather is much kinder and gentler, we had 100% survival of our drone colonies, so we had polyandry at level 9 males or 54. So there's nothing magic about these numbers. These numbers meant that I had 9 drone source colonies that survive. Colonies that received low polyandry received 1 male from each of those 9. Queens that got high polyandry received 7 males from each of those, 6 males from each of those colonies for a total of 54. And then again, those drone lines were either previously selected for Varroa sensitive hygiene or not selected. Here we are the day we set up this experiment. That's Debbie on the very far back left. That's Kirsten Trainer on the far left. Kirsten just opened a new magazine, A Million Blossoms, I think, and I think she's currently living in Germany, though she is American. And that is my sidekick on the back waving her white shawl, Jennifer Berry, whom I think you all have met and has spoken in the UK before, as has Debbie. And we put them in a circle like this just to kind of minimize drift and any spurious location effects that may be going on. And so then we tracked these colonies for the measures of fitness that I'm going to show you now. Okay, let's talk first of all about the overarching effects of polyandry. This is just looking at polyandry averaged across the semen types, just the overarching effects of polyandry. Let's start at the top left. In Delaware, we had a significant increase in brood as we moved from low polyandry to high. In Georgia, we had a significant increase in bee populations as we moved from low to high. In comb construction in Georgia, bottom right, we had a significant increase in comb construction from low to high. And in Delaware, also comb construction from low to high. So this doesn't take an Einstein to interpret these data. I mean, polyandry rocks. And I think this is worth pausing to just linger on a second to think about. These averages that you're seeing right here are irrespective of selection. You know, this is averaging across wild type drones and VSH. And I find this interesting to think that without any selection at all, I mean, zip, nada, with no selection whatsoever, we can get these economically important characters just by increasing the mating count in our queens. And those of you who have worked in breeding, I mean, you know, and I know how difficult it is. It's not a cakewalk. You have to select whatever character you're going for, which means you have to actually measure things. It takes time. It's hard to do. And then you have to propagate from using the criteria that you established. You got to rear these virgins, and then you got to mate those virgins. How are you going to mate them, right? That's a big problem. And then you got to wait for the next generation to make your next selection choice. So it is inherently slow and laborious. And I'm sitting here looking at data like this, and I'm thinking, and all you have to do is just pump them full of drones. Incidentally, I got to tell you a story. I was talking about this. I was talking about breeding and polyandry and some earlier experiments we were doing. And I had a gentleman in your country. I think he was with the London group. He was in the metro area of London, I remember. And this little guy, he piped up and he says, "But won't she pop?" And I thought, you know, he's got a good point. You know what? But what we're doing in these studies is we're normalizing the sperm dose. We take the sperm from X number of males, and then we literally mix it in a little Eppendorf tube with saline and a little glass rod. I mean, we're mixing it all up to make it as homogeneous as possible. And then we take a standardized dose from that mixture. So the dose is always the same. So it really is just drone number. It's not semen volume that we're talking about here. So look, voila, great results with just increasing mating number. Okay, let's take a look now at the overarching effects of genetic selection, whether that semen came from VSH stock or whether it came from wild type. It's not quite as interesting, is it? Top left, Delaware brood, no effect. Georgia populations, no effect of prior selection. Comb construction in Georgia, bottom right, no effect. In fact, the only effect we found between genetic types was with comb construction in Delaware, and it was in favor of the wild type non-selected drones. Now this should not be terribly earth-shaking news because these are VSH drones. We have no expectation that they're going to be good comb constructors, okay? So that's not really a big deal, but I do think it points out, it is consistent with that premise I told you earlier about genetic bottlenecks. In this case, you know, it kind of looks like VSH in Delaware, you know, suffered in its ability to construct comb compared to the non-selected drone semen. So there it is. This is a non-controlled piece of evidence. It does superficially support what I'm saying about genetic bottlenecks. But obviously, we'd have to design a much more robust experiment if we really wanted to chase that rabbit more. But this is at least consistent with that point. Now, mites. It's really all about the mites, isn't it? Well, here we go. In Delaware, moving from one to, rather VSH or wild, we had no effect. Georgia, VSH or wild, no effect. It's rather grim, wouldn't you say? This kind of gets back to a comment that I made earlier about just how hard it is to cram Varroa resistance into a bee that has no genetic tools available to it specific to this pest. So VSH is inherently a tough nut to crack. However, let's add things to this and look at polyandry. When we added polyandry to the mix, in Georgia, and only in Georgia where we had the higher levels of polyandry, we found the VSH phenotype only expressed at the highest levels of VSH, where we did have a significant reduction in colony mite levels with the highly polyandrous stock. This is a positive synergy between previous efforts at selecting for Varroa mite resistance with polyandry. Now, there is some theory that did not originate with me, but it is some theory that originated with Rob Moritz in Germany and Stefan Fuchs. And they were anticipating results like this with a piece of theory that they published in 1999. And they said, we predict, again, there weren't data yet, it says, we predict that high levels of polyandry will in fact be adaptive for extremely rare behavioral traits that are beneficial. They predicted this very thing, which I find pretty exciting. And the point, their rationale goes like this. Okay, think about it. A specialist is by definition rare. I mean, duh, that's what a specialist is. If you had a bunch of them doing the same thing, they wouldn't be specialists, right? They would be generalists. So you got a character here that is rare, and it's adaptive to be rare. Because if you had an entire colony that did nothing but remove brood, it wouldn't go far, would it? This would be maladaptive at extreme expressions like that. But if it is rare, then it will be adaptive, okay? Number two, if it's rare, then how is the colony going to get it? And this is where it interfaces with our results. They're going to get it by the queen practicing extreme levels of polyandry. So it is a strategy for having your cake and eating it too. She needs to mate with enough males that she captures one or two of them that have the important allele that I need for my colony to survive. And let's say that I got that male on my very first copulation. Well, I can't stop, can I? I have to keep on mating in order for that effect to be rare in my colony so that it's not overexpressed. So this was the theory that anticipated the results that we are showing in this experiment. And I find it really exciting because it interfaces biology with what queen breeders are already doing. And I am proposing here today that we need to be more purposeful about integrating high levels of polyandry into our breeding schemes that are doing a very good job at discovering and augmenting in selected populations very, very specialized character traits. Hygienic behavior is one of them. Grooming behavior is another one of them, where workers can detect mites on themselves or on their sisters and pick them off and bite them. So we want these genes. We want to be able to put our fingers on them, okay? We want to have, we applaud the efforts of people out there that are doing these laborious selections to winnow the field of Apis mellifera genome and concentrate these known alleles in known germplasm. We want that. That is a good thing. But our data are indicating that that needs to be interfaced with high mating numbers in order to exploit this principle that Moritz and Fuchs discovered back in 1999. I think this is pretty cool information and warrants pretty loud sharing. Right, so how does it apply to the real bottom line? Okay, what really matters in biology is survival. And by God, we measured it. Here you are looking at our survival ratios. Now, these are just data for Georgia, but these are frequency distributions of colonies that survived until the next spring. The red area are the survivors. The blue area is the dead outs. So red is good in these graphs. And again, it mirrors the results that I already told you about mites. We found overall higher survival in the VSH lines in Georgia. We found no direct effects on survival with polyandry. But when they were integrated, we had the highest survival in those colonies that were both VSH selected sperm inseminated to 54 males. So this just mirrors perfectly the data that I showed you earlier on mites. Now here's something else interesting. It does not mirror the results on comb construction and bee population size, both of which were higher in the colonies that had inseminations with 54 males. So it appears that in the case of a really potent environmental stressor, such as Varroa destructor, that any benefit that we gain from polyandry toward that one stressor eclipses what other benefits may have been gained in other character states that were not so critical to the survival of that colony. So in the case of Varroa, you get survival where it counts with polyandry and prior selection of Varroa resistance alleles. I think these are important results. I think these are good data for queen breeders to have in hand. And I think a very natural question that should be on all of your minds is, okay, so what? How do we get more polyandry, right? Well, the first and easiest answer to that is instrumental insemination. You know, it works. You know, we've used it now for quite a while. Giles and I used it in 2012 and 2013. We've used it again with this. We're continuing to work on it with experiments we have coming up this season. So, you know, instrumental insemination works. I have one of my master beekeepers who emailed me the other day, and he says, do you know that you can 3D print insemination devices? I said, no way. And he says, yeah, here it is. And he sent me the link for $15. You can follow this link and print your own instrumental insemination device. So there you go. With technology like that, it brings at least the price to a point that's not an objection. Now, there still is a learning curve. You know, there's workshops to attend, there's practice. It is a bit of a thing to learn, but you know, people do it, and more people are doing it. I had the good fortune to visit Poland back in 2005, I guess, and I got a tour of the resources for beekeeping in that country and was really impressed at the state-sponsored queen production labs that they have there, in which production queens are inseminated instrumentally and then shipped out to beekeepers. So in Poland, they have elevated instrumental insemination to a high pitch of perfection, and they function just as well as naturally mated queens. We're getting better here in this country. Our commercial breeders in California are using more and more insemination, usually in the context of preparing breeder stock. But it is a slow wave building momentum, and I think that should only increase. We are also looking at a proxy method, and this is data we just got in, we finished last year. We haven't even analyzed the data yet, but we're trying to do an end run around polyandry and accomplish the same thing by a very, very beekeeper-friendly method, and that is swapping brood. Think about it. This is something beekeepers already do. You know, we move brood around, we're making splits, or we're equalizing colony strength. So this is not a strange concept. You know, we'll take sealed brood from one colony, stick it in another. We do this all the time. Well, think about it. When you do that, you are causing a temporary flush of polyandry in that colony. Depending on the mating number and depending on how many colonies that you put into one colony, you could very quickly achieve huge polyandry counts. So we did a 2-year replicated study where we did this. And through this brood swapping and sharing scheme, we were creating temporary bursts of polyandry in the neighborhood of 70 patrolines expressing in a colony at one time. Now, granted, this is a temporary effect. You know, you get this flush of polyandrous workers, and then unless you keep up the scheme, which I would not recommend doing because it's a lot of work, eventually that cohort is going to die off and you're going to get back to just whatever the polyandry of the resident queen is. But for that time, you have whatever benefits are available to you of polyandry. So we have the data in the bag. I can't even tell you what the averages are. That's how bad we are. We haven't even looked at the data yet, but that's next on our list to start writing those data up. And then finally, I think we need to revisit the idea of drone concentration in our mating yards. We know, for example, that you can get desirable matings, and by desirable, I'm thinking in the context of research done at USDA in Venezuela back in the 1980s, where the whole thing was Africanized bees. And USDA was being heavily funded to get a handle on this bee before it arrived in the United States. And so we were very interested at that time in trying to get European matings rather than European African, that made a very defensive hybrid that was obnoxious to work with. And they were able to show through extremely high rates of drone saturation in Venezuela that they could get some degree of European matings. So we know that we can influence matings, but we don't know if we can influence polyandry by increasing concentration of males. And then finally, I think this is my favorite one of all, I wonder if we can select for this. I honestly wonder if we can select for queen promiscuity and just leave it to some Texas or California breeder to come up with a really catchy trade name. But it's going to happen, I promise you. Someday someone is going to select for a promiscuous queen. And maybe it is heritable, and maybe it would give us the benefits that we want. So these are some ideas that we're chewing on to translate this into principles that beekeepers can put to good use. So let me kind of wrap up my points for you here this evening. I'm really rocking on this idea of a European origin of the genus, simply for the reason that it provides satisfying answers to what I consider two of the biggest puzzles in the biology of the honeybee today, the divergent distribution between Serrana and mellifera, and more importantly, why mellifera has no genetic tools whatsoever for dealing with Varroa. The experience of the mellifera lineage in Africa has shown us that it can occupy virtually any terrestrial niche on this planet. If there's flowers that bloom, mellifera can colonize it. Apis mellifera's mating biology is radically oriented toward within-nest diversity. And I talked about the many levels by which this species tries to achieve that, through the formation of its gametes, through drone congregation areas with many colonies represented, through the queen's promiscuity and her spermatheca. It all accrues toward genetic diversity. And contrast that with human-oriented schemes where we try to do the exact opposite, and that is to narrow and constrain them toward a particular character of our interest, very likely losing some other important genes in the process. We have shown, and others have shown, Giles and I are far from the only authors in this field. It's a very important field right now. Many authors have shown that high mating numbers improve comb construction, bee populations. We have shown before brood production is increased, foraging is increased, the efficiency by which workers recruit one another in foraging dances is increased. I mean, it's lots and lots of good stuff that happens with a colony whose queen is multiply mated. We have shown in this study that high mating numbers synergize positively with prior selection of rare resistance genes. And it seems like with a really potent stressor like Varroa mites, that this benefit can actually eclipse and supersede other benefits and overexpress it in favor of that one really potent stressor, as we showed Varroa mites.
Keith, you say you need to have plenty of drones around for mating. If you want 200 queens a week mated, how many drone production colonies should you have within a mile radius?
I don't have a formulaic answer for that.
Or a guess would do.
Yeah, I would, and I would not, one thing I would say too is I don't think you have to worry so much about a mile radius. I would try to get them in there as close as possible. and I know there has been some confusion in the literature over the years, and I think it's even out there. You can find papers to this effect that you should have the drone colonies X number of meters or miles away from your mating yards. Otherwise, you'll get unsavory pairings and brother and sister and that kind of thing. I wouldn't worry 2 minutes about that. I would offer instead that polyandry is yet another safeguard against those close matings that probably do occur. You know, that we have no reason to think that a queen can avoid mating with a brother. So it probably does happen, but I would say polyandry is sufficient to negate any of those mismatches that may occur. Ged, I don't have a good answer for you. I think a lot. How's that? And we've not done those experiments yet. We've done the brood mixing. We've not done the drone saturations yet. I could envision a day when we are trafficking in drone semen. For example, what if here's kind of like the next step. You know, we have shown really good results and really good synergy with one selection character, that's VSH. There's others. What about groomer stock? So I could see us really front loading our queens with characters that we are pre-selected, we know that we like and are good, as long as we keep in mind that those have to be rare. I know it's paradoxical, but it makes sense when you think about it. You don't want a colony full of specialists. You just want some specialists. And I could seriously envision us getting to the point where we can customize these capillary tubes and ship them off to breeders. And where you're getting these optimized mixes of rare characters and high mating numbers. I think that's very within reach. We could do that. But yes, trafficking in drones, I think, is very natural outcome of this study. You know, that's a good question, and there is a reason why that question keeps coming up, because the literature has evidence of both. Okay? If you look at some of the older literature, there is some examples of stratification, but I can strongly say, and I say this strongly because I've read this literature and summarized it. The vast majority indicate that it is not stratified, that it is in fact a random distribution of all of her mates at any given time, which also makes sense. You know, doesn't it? It makes sense. If this is true and really adaptive, then it would counteract the adaptation if that semen were layered. So you only get the benefit of it if in fact you are having a general representation of all of your drones at any moment.
We quite often get overwinter supersedias. Are they likely to be matings within the apiary, or possibly matings within the hive? I mean, does that ever happen? And is that like a last-ditch attempt where the queen's dying out to just continue the line rather than select for diversity, as you say? Any thoughts?
Yeah, I do. You've tapped into something that deserves its own lecture, and I promise I will not do that now. But I will give you the real gist of it. The latest thinking about but first of all, let me say, what you propose is in fact true. I mean, it's all things equal, it's better to raise a queen, even if it is out of season. It's worth trying. So there's nothing ventured, nothing gained. So ecologically, that makes sense. But at an evolutionary scale, there is 2 papers that have been published fairly recently that propose that supersedure queens are in fact parasitic. And it works like this. Think about a normal queen rearing situation. Who is in charge? Who chooses the next queen? Well, actually, it is the old queen. And the way she does this in a normal swarm sequence she will deposit eggs in queen cups. And these are, little cups along the, and we know that statistically these eggs deposited in queen cups have a higher than average chance of actually being reared into a queen. So this is a very low level effort of the mother to determine who needs to be the next, who gets to be the next queen. And then the swarming happens and the next queen takes over the nest and voila, it's done. Okay, now in the situation that you describe in a supersedure, who calls the shots? If it's off season, there's no reason whatsoever to think that there's any cups with eggs in them. In those situations, it's the workers who determine who gets to be reared into a queen. And once again, Robin Moritz, this name keeps coming up. This guy is unbelievably good. But Robin Moritz published this paper and where they showed that there are in fact patrolines that are sired by particular drones who are liable to become queens. And imagine, here it is, you got this royal larva and the colony has gone queenless. Boom, oh no, mayday, mayday, we need a queen. And here's these royal larvae who essentially say to the nurses like, feed me royal jelly. Hey, you up there, royal jelly here, not there, here.
Wow.
So they are biasing the behavior of their nurses, probably with some kind of pheromonal cue. That is interesting, but it gets worse. These royal patrolines are virtually non-existent among the workers. They don't rear them into workers. So it's as if these larvae are saying, hey, you up there, either feed me royal jelly or don't feed me at all. You can imagine how melodramatic that sounds when they say it. But that's essentially what's happening. So you have this patroline that has no representation among the workers, and it's only represented in queens. And it appears to be a bid to get around that whole control thing and to take charge. I want to be the queen. So it is sort of a dissolutive effect in evolution and kind of a step away from the superorganism, an attempt to go back to selfish behavior. Because once an individual goes toward the path of reproducing, they don't do any work. We see this in these patrolines that don't even bother to make workers. And we also see it in laying workers. When the queen's pheromone is removed from a colony and her ovaries develop and she lays eggs, those laying workers abandon doing any colony work. It just seems to be a rule of biology. If they reproduce, they're not going to be altruistic workers. And so the colony goes to pot. So this, we think, is perhaps an explanation for why supersedure queens tend to not be very good. They're raising workers that are not good at working. And traditionally, we always ascribe that to other reasons. We say, oh, well, gosh, the supersedure, it's reared in the off season. They don't get fed as well, and they're crappy queens and wah, 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 which may be true. But this alternate explanation, I think, is a little more satisfying. The average, I can give you a number. The average is 12. 12. That is based on work by Dave Tarpey, my colleague next door at North Carolina, NC State. And Dave did a meta-analysis of all of the mating numbers of mellifera in the literature, and it's right at 12 is the average. The highest in the literature, again, comes from Dave Tarpey's lab. One of his students published a number of 77 males in a natural mating situation that they were able to discover. So the numbers skew really, really high, but the average is way down here between 12 and 20. And I think there's some understandable reasons for that. It is not risk-free. You risk birds, you risk sexually transmitted diseases. So an infinitude of mating is risky. But I'm wondering if we could push that in managed systems. Drones are where mites reproduce, And one way to control mites is to purge our colonies of drone brood, and in fact use drone brood as a trap and then freeze it and kill it, which works, by the way. It works. You get significant reduction in mites. I say this is one of those tensions where these two principles are working opposite one another, but I tend to be pretty high charged on polyandry, so I'm going to tell you, forget about the mites, fill your colonies with lots of drones, probably 2 or 3 combs of drone brood in the interest of getting these polyandrous queens, at least in the queen rearing season. And then we can cut back if we wish later in the year and practice our drone brood trapping methods. So I think it can still be accommodated. If the characteristic is heritable, and characters vary in how responsive they are to selection, either natural or artificial. I think it's a very attractive idea, and I would love to test that hypothesis. But if it is heritable, then yes, one would think that her daughters carry it, because it would be a maternal character that all of her daughters would possess. So I would think, yes, that would be a natural thing to pursue.